Today we are going to look at the visual pathway. What does that mean? Well, that means neuroanatomy for one thing. That means we're going to look at the pathway, the route the neurons take that carry light from the retina in our eyes back to the occipital lobe in the posterior part of the cerebral cortex. There are a number of parts to it. We'll look at the parts, we'll look at some physical things, but we're going to have to draw this out, those neuronal pathways. The reason it's interesting is because there is some crossing over to other sides of the brain, but some things stay on the same side, and injury to different parts of that pathway will have different effects upon vision. So we'll try and work all that out, okay? Is the creche behind the anatomy lab having playtime? Yes, it is. You might be able to hear that. Enjoy. First things first then, visual field. What is the visual field? So the visual field is what an eye in a fixed position can see. But there's more to it than that. You need to do something here. Okay, so I can see both of my hands. Now you do the same thing. You do the same thing, especially if you're on the bus or on the train. Now, if you close one eye, I'm closing my right eye. I can't see that hand anymore. If I close my left eye, I can just about see my thumb there. So each eye has its own visual field, and those visual fields overlap. So if I close that eye, I can still see on the nasal side, but I can see a really long way laterally on the temporal side. If I close this eye again, I can see a long way temporally, and then as I move my hand nasally or medially, I'm going to lose it about there. So each eye has its own visual field. Those two visual fields overlap to give us binocular, stereo, 3D vision, so we can determine how far away something is. And the visual field has lateral parts and medial parts for each eye, which might be called nasal parts and temporal parts. You can see your whole field of vision. Close one eye. What do you lose? Close the other eye. What do you lose? But what do you retain? So it doesn't matter which eye is open, you can still see this bit in the middle here. You just lose this lateral bit. Okay, now I'm going to illustrate this by using this bit of video. So if I have both eyes open, this is what I can see with this bit of video. If I close the left eye, I lose that part of the frame. If I close the right eye, I lose that part of the frame. So I'm not going to make it blurry, I'm just going to make it black. So that's what we're seeing, whereas that central part there is visible to both eyes. And as we go through the visual pathway and we cut various bits of it, I'll use that video to demonstrate the parts of the visual field that are lost. Next lesson, the shape of the retina adds even more complexity to this. Check out this spoon. I'm just tilting the spoon a little bit, but what can you see? What does the world look like? Does it look the right way up, or does it look upside down? It's clean at least. Hasn't got as much coffee on it as it normally has. I'm now behind the camera, holding the spoon. Is the world the right way up, or is the world upside down? And if it's upside down, is left on the right and right on the left? Yes. So this is a concave shape, which means that the light doesn't come into it like a flat mirror and bounce straight back at you. It comes in, it bounces back at a different angle. The crux here is that the concave spoon, the image that it collects is upside down relative to the world. The retina has that concave shape as well. Let me get a model. Eye, oh, it comes out, jolly good. So the eye is it's a globe, it's a sphere. If I take it apart, we've got that same curved surface that the spoon has. So the light collecting surface is over here, which means that the light that's collected on the retina is going to be upside down, and left is going to be on the right, and right is going to be on the left. Just remember the spoon as we draw our diagram, okay? Now we've got enough information to start drawing our visual pathway. Drawing, not my strong suit. Okay, so we've got curved retina. And I need 4 colours. Hopefully these are going to be distinct enough. So if we're, now those visual fields, I'm going to over-egg them. I'm going to say over here we can see this colour. And over here we can see this colour. I'm exaggerating kind of the fields, but to help you understand the principle. Right, so we're looking at these colours ahead of us, right? This is a retina, and this is a retina. So this means that if you remember the spoon, the light from here is going to come in here. The light from here is going to come in here. And the light from here is going to come in there. And the light from there is going to come in there. And the retina is a collection of light detecting neurons that's going to detect that light and send it back to the brain. So this light centrally is captured by the lateral parts of the retina, and this lateral light, right, these lateral bits of vision are captured by the medial parts of the retina. Now these neurons that are collecting light on the medial parts of the retina will cross to the other side of the brain. But the light that's being captured on the lateral side of the retina will pass to the same side of the brain. So this is the first neuron. These are passing to the same side of the cerebral cortex. Remember the colour red. Okay, so the lateral stuff is, again, it's staying on the same side of the brain. So it's not a case that all of the light from the retina passes to the other side of the brain, or all of the light from the retina stays on the same side of the brain. It's worse than that. Some of the light crosses to the other side of the brain, some of the light stays on the same side of the brain. So what have we made here? This is the optic nerve, and this is the optic nerve. And we'll call this the left side and this the right side, okay? So the optic nerves from each eye run back inside the cranial cavity and they cross here. So this is the optic chiasm. Optic nerve, optic chiasm. Now this bundle of neurons then enters the brain, so it becomes part of the central nervous system. It's still a bundle of axons, a bundle of fibres. It's just that when it's in the central nervous system, we change its name again and it becomes the optic tract. But this is still the first neuron. And these fibres are running to the lateral geniculate nucleus, or the lateral geniculate body, which is part of the thalamus, or certainly next to the thalamus. Whenever I'm teaching the thalamus, we talk about the lateral geniculate body and the medial geniculate body. So we've created optic nerve, optic chiasm, optic tract, and down here we're at the lateral geniculate nucleus, and this is where synapses are occurring. If we go back to our model of the eye, there's the optic nerve there, leaving the retina. So that's the optic nerve there. Those two optic nerves, here's O'Brien, those two optic nerves run together and they meet at the optic chiasm where they cross over. Now the optic chiasm sits just above the pituitary gland. So there's the optic chiasm there, and you can see it. So you can imagine that the cut end of the optic nerve is here. So we've lost the actual optic nerve because we've cut that to remove the brain from the skull. Obviously the eye stayed in the orbit. And then the optic chiasm continues as the optic tract. And we can see the shape of the tract as it runs into the cerebrum there. Now the brain falls apart when I try and take it apart. But yeah, what we're seeing there is we're seeing the optic tract running around to the thalamus, which is in here. The thalamus is very central inside this, in the centre of the two cerebral hemispheres. They're kind of overlapping over it. So they run back to the thalamus. Okay. So now these will synapse with a second neuron, and these neurons will all run back to the occipital lobe. So the occipital lobe, the visual cortex is back here. And this is very well organised, but we're not going to talk about that today. We're going to talk about these roots here: optic nerve, optic chiasm, optic tract, and what have you. All right. So what happens if these things get damaged? First of all, what happens if you were to just cut through the optic nerve? You'd have a complete loss of vision in the right eye. Left eye would obviously be fine because it's still sending its neurons along. So here we'd have loss of vision in the right eye, and if we go back to our little bit of video, that would look like this, kind of, probably. What about here? What if we cut through the optic chiasm? What would you lose then? Well, okay, follow the blue. So you'd lose the medial retina image, which is actually from the lateral part of your field of vision. So you'd lose the lateral or temporal field of vision in the left eye. Remember, we're following the colours, blue. And we'd lose, same thing on the right eye, we'd lose the lateral field of vision from the right eye because there's the purple. They wouldn't be able to, yeah? So what would that look like? So this means that if you're walking along, walking through doorways and what have you, you're less likely to see the edges of the doorways. So people might complain of, oh, I keep banging into the doorway, or I keep banging my car, I keep pranging my car, because they've lost their temporal fields of vision or their lateral fields of vision, but they haven't really noticed. So on our illustration, this would kind of look like this, right? I've said these lateral fields of view are temporal parts of your field of view, hence because it's both eyes, temporal, bitemporal. Anopia or anopsia means a loss of vision. So hemi means a half loss of vision in each eye. Now this is known as bitemporal hemianopia or hemianopsia. Bitemporal hemianopia. So you would lose that lateral. Now the reason this is interesting is because, as I said, the optic chiasm lies superior to the pituitary gland. So if we grab our skull, take the calvarium off, so there's the orbits. Inside the skull, this here, that depression in there, that's where the pituitary gland lives. And look, it's surrounded by bone except for the superior part. And superior to there lies the optic chiasm, which we see on our brain here, which means that a tumour of the pituitary gland sat in that bony depression means it can't extend inferiorly, so it must push superiorly. So a tumour that expands in the pituitary gland will push up into the optic chiasm and could lead to this and this bitemporal hemianopia. One of the reasons the anatomy of the visual pathway is so interesting and useful clinically. Okay, where's my pens? What about here? So, assuming these things are all intact, if the visual tract was injured, we would lose, okay, where's the red line go? We would lose that part of the retina, So we'd lose this right nasal field of view. And where's the blue one come from? Blue one comes from the medial part of the retina. So you'd lose the left lateral, left temporal part of your view. So you would lose your vision or lose these partial fields of vision. That is known as, or it's on the left side, so that would be left homonymous hemianopia. So hemianopia again, because we've got half a loss, half a field of vision lost in each eye, it's the left sides that are lost. So left, so homonymous means, you know, two words that sound the same but mean two different things. How does that help? I don't know. Anyway, so left homonymous hemianopia if you cut through the optic tract. I mean, what happens if you were just to cut through that bit? Ooh, four, bump, bump. So not the optic chiasm, but the fibres that are running, they're staying on the same side, right? So what happens if you were to cut or injure or compress or damage these fibres? Well, that's coming from the red bit of the retina here, which is coming from the right eye on the nasal side. So you can work this out, can't you? Once you've drawn your illustration, you would get this. I don't know why I've drawn a line in the middle there. There we go. The vision in the left eye is just fine. That then, that's another hemianopia. It's in the right eye, and it's the nasal field of vision that's lost, so that would be a right nasal hemianopia. Now that really is most of it. If you can remember the retina, the spoon, this diagram, you can work this stuff out. There is another weird one which might be cool to mention. This one here. Okay, so if you were to damage the these fibres running from the lateral geniculate nucleus to the visual cortex and the occipital lobe, something weird can happen. Now obviously it's going to be the same as 3, right? Because these are the same fibres, they've just synapsed through the lateral geniculate body. But what you get is you get the same sort of thing, but you get sparing of your macular vision, your central vision, your high def vision. Now the reason this might occur is because most injuries to these tracts of fibres are likely to be caused by ischemia, so blood not getting to this region of the brain, to these tracts. So the blood to the occipital cortex is largely supplied by the posterior cerebral arteries, a little bit by the middle cerebral arteries. So injury here is likely to be caused by clots blocking the blood vessels, causing again the same sort of symptoms as we saw here. So left homonymous hemianopia, but with macular sparing, because the fibres that carry information from the macula, the really high resolution parts of your retina, they get a bit of a dual blood supply. There's a bit of an anastomosis in there. There's a bit of a backup blood supply. So if it's an injury to the blood supply that's causing this, then because of that overlapping blood supply, it's likely that the nerves that are carrying information from the macula in each retina will still present that information to the occipital lobe just fine. So you might come across this, but you know. So that's the crux of it. These are the visual pathways. Remember, the spoon means that everything is left to right and right to left and upside down. And then remember that the lateral part of the retina, those neurons stay on the same side. The medial part of the retina, those neurons cross to the other side with both eyes, giving you this diagram. Try drawing this diagram. You should sit down and draw this diagram by working this out, which means that those two things overlap, means that if you sever the optic nerve, you lose vision in one eye. If you sever the optic chiasm and only affect these fibres that are crossing from side to side, then you get this effect. bitemporal hemianopia. And if you sever the optic tract, where half of the fibres have come from one eye, half of the fibres have come from the other eye, and because of the spoon, meaning these things are left to right and right to left, just to make everything even more complicated, that means that you'll lose the visual fields, like you'll lose the left visual field in each eye, giving you left homonymous hemianopia. You have this possibility here where you just affect one eye, and if you affect that same tract essentially after it's synapsed with the lateral geniculate body, because of the extra redundant blood supply that the neurons carrying information from the macula get, you might get the same thing but with macular sparing. It's fascinating, it's cool, it's really amazingly laid out. I love looking at this sort of thing. I just hate doing this on video because trying to film a whiteboard and trying to make it clear on a whiteboard is just, it's horrible. I hate it. But there you go. These are the visual pathways. And that's what happens if it goes wrong. And that's why. So it's worth knowing, okay? The eye. Very complex. Okay, I hope that was vaguely clear. See you guys next week.